Do you know genomic imprinting? It has been reported that imprinting is a particularly important genetic mechanism in mammals, and is thought to influence the transfer of the nutrients to the fetus and to the newborn from the mother, and affect the growth in the uterus and behavior after birth. Aberrant imprinting disturbs the development and is the cause of various disease syndromes. Now, let's recognize what genomic imprinting is. Genomic imprinting is defined as the differential expression of a gene or chromosomal region according to parental origin of the inheritance. Both the maternal and paternal alleles are present, but while one is functionally active, the other is inactive in somatic cell. Please look at this figure. Gray allele is non-imprinted, and here paternal allele is imprinted, and for this one, maternal allele is imprinted. And in 1984, two researchers independently published the results of the experiments on newly fertilized mouse eggs. First, remove fertilized mouse oocyte from the ampulla of the oviduct as the pronuclear stage of development. And then, remove either the female pronucleus or the male pronucleus with a very fine pipette and replace with a pronucleus of the opposite type, thus producing a diploid zygote. And then implant the zygotes into a pseudopregnant female mouse. They had generated embryos that contained either two sets of chromosomes inherited from the mother or two sets of chromosomes inherited from the father. However, when the embryos are transferred into pseudopregnant recipient females, the embryo failed to develop to term. When an experimental zygote containing two male pronuclei is implanted into a pseudopregnant female mouse, it develops as a trophoblast and gives rise to a mass of the placental membranes. Very rarely, an embryo forms and develops to a stage comparable to approximately a 3-week stage of human development. In contrast, zygotes containing two female pronuclei develop as small but recognizable embryos with reduced placental membranes. These remarkable results sent a clear message. Despite being genetically equivalent, the set of the chromosomes inherited from the mother were not functionally equivalent to the set inherited from the father. For normal development to occur, one set of chromosomes from each parent was required. Then what are the genetic features that characterize imprinted genes? Genetic studies showed that imprinted genes were not evenly distributed across the whole genome, but located in particular genomic regions. Thus, one remarkable and characteristic feature of imprinted genes is that they are rarely found on their own. Around 80% are physically linked in clusters with other imprinted genes. For example, here shows human chromosomes 11. Red labels maternally expressed genes, and blue labels paternally expressed genes, and black labels biallelic expression. These imprinted genes are linked in clusters just like this. Furthermore, two general features of the DNA sequence and environment of imprinted genes have been noted. First, they are usually rich in CpG islands. Approximately 88% of a mouse imprinted genes have CpG islands. Second, the great majority of imprinted genes show differences in DNA methylation between parental alleles. And here highlights the allele-specific epigenetic changes, such as nucleosomal condensation through decetylation and methylation, and opening of the chromatin by acetylation and demethylation. It has been showing considerable changes in methylation during the development and acquired tissue-specific methylation patterns, which can be associated with the tissue-specific imprinted expression. And the genomic explains change in characteristic ways during the life cycle of the organism. The germline has the role of resetting imprints such that in mature gametes. During aerasia, there is apparently genome-wide demethylation in germ cells. Imprints are established during the development of germ cells into sperm or eggs. After aerasia, methylation begins in both germlines at late fetal stages and continues after birth. Oocytes are in meiotic arrest and methylation occurs during their growth, whereas during the spermatogenesis, methylation occurs before meiosis. After fertilization, they are maintained as chromosomes duplicate and segregate in the developing organism. In the germ cells of the new organism, imprints are erased at an early stage. This is followed by establishment again at the later stage of the germ cell development, thus completing the imprinting cycle. And next question is, what mechanism underlies the independent expression of maternal and paternal genomes during the early development? Over the years, many investigations have uncovered regulatory relationships between DNA methylation, histone modification, long non-coding RNAs, and associated proteins. After class, please search more papers about the mechanisms underlying the independent expression of the maternal and paternal genomes during the early development. Well, 36 years after the original discovery of the genomic imprinting, where do we stand now? Though we still don't understand the evolutionary processes that have led to the emergence of the genomic imprinting in mammals, it has revealed a wealth of the epigenetic insight that continues to have an enduring influence on genome biology. The advantage of the genomic imprinting may be effectively prevent sexual reproduction and maintain genetic diversity in the history of the evolution, but increase the risk of the recessive mutations to dominant. Well, this is the genomic imprinting. Thanks for listening.